So this is the second lecture on genome annotation from my bioinformatics class. Just wanted to preface the lecture by saying a lot of the information, or a good portion of this information and some of the slides came from a presentation that I was able to co-opt information from my old postdoc advisor, Rachel O'Neill. So a shout out to her. She's a professor at University of Connecticut and a big influence on me. So moving on, today we're going to be talking about how annotation intersects with ontology. So we're going to talk a little bit about what ontologies are and how we use them, specifically how we use them in biology to infer important parts of biological function and roles with regard to what they're doing on genomes. We're also going to further drill down on this idea of like what's the difference between the structural and the functional based annotations. We talked about structural annotations last class, and we'll talk about functional ones today. And furthermore, we'll go into a little more detail about what the gene ontology is, how it's structured and organized. So we talked last class about annotation, right? How that an annotation was a way of offering some sort of explanatory note or commentary on something. And in this case, we're talking about annotation of the genome. So we're talking about, what are the metadata that we can associate with specific strings of nucleotides. And last class, we spent a lot of time talking about how we can predict elements based upon rules. And today we're going to put more emphasis on how we actually attach biological information to these elements. So last class we talked about structural based annotations. Today we're going to talk more about functional annotations. And so in this case, what we're particularly interested in are what regions do, and in other words, like what are they actually coding for. So in this case, a lot of the things that we're identifying using a homology-based approach, and how we're actually assigning the function in this case is under the assumption that because they are homologous or similar in their molecular structures, they're going to have a similar genetic function as to other that we would have described in other genomes. To further characterize these types of structures, we're going to use an entity called the Gene Ontology, which helps us have a common language for defining what these things are. There are lots of other examples, but this is also the way that we would also identify things like orthologs and paralogs. So a couple examples we're going to be using when you do this type of work. You're very much reliant upon information that is already existing in databases. And so these are some of the main databases. They're not all of them by any means, but some of the principal players in this field of annotation. There's the Gene Ontology, which is one of the most well-known databases for annotation. There's the enzyme code repository. This is run through the European Union and by the International Union of Biochemistry and Molecular Biology. They're the KEGG maps. This is a Japanese-based database, and these typically represent a very high-level curated pathway map of the knowledge of how molecular interactions and reactions are interacting with each other. So a lot of metabolism in here, enzyme-oriented activities. Interpro is another European database, and this is one of the main ones for retaining information on protein signatures or protein families. So how we identify the different components of proteins and whether or not those domains are shared among proteins is often a good indicator of whether or not they belong to the same family of proteins. And finally, there's the protein data bank, and one of the unique things about this is that there's a lot of three-dimensional structures that are retained in this database. It's a US-based one. So you might be asking the question, what is an ontology? Because I bet a lot of you may not be all that familiar with this term. So ontology, back in 1721, Bailey defined it as an account of being in the abstract. In 1733, there was a brief scheme of ontology, or also the science of being in general. In 1832, we had Bentham working in this field. So the field of ontology, or as it may otherwise be termed, the field of supremely abstract entities, is a yet untrodden labyrinth. Wonderful bit of turn of phrase there. And Bosanquet in 1884 said, ontology as a doctrine of being and relations of all reality had precedence given to it over cosmology and psychology, the two branches of inquiry which follow the reality into its opposite distinctive form. So I think it should be pretty obvious what ontology is by this point, right? No, but you can see that as this term is coming into the lexicon and is being used, it's this idea of understanding what things are, right? What is the state of being of these things? What are they? As how do we take these abstract things and put them into some sort of less abstract entity? So ontology was big in the area of philosophy, and it was identified as a specific branch of metaphysics, right? And so, which concerned the nature and the relations of beings. So if we think about what metaphysics is, right, we kind of know what physics are, trying to understand the fundamental nature of something. And the metaphysics, we're actually understand the fundamental nature of being. And so the physics of physics, if you want to say, like the metaphysics. So the fundamental nature of being and the world that it encompasses, right? And so if this is a branch of that, right, ontology is trying to figure out what is the nature and the relationships of the things within it. So some of the big questions in metaphysics, or if you think, is what is there? What is it like? And Barry Smith, I kind of like this quote, which is that science, what is an ontology? Is the science of what is, of the kinds and structures of objects, properties, events, processes, and relations in every kind of reality, right? So very fancy stuff. A more recent definition by Gruber discusses how ontologies provide controlled and consistent vocabularies to describe concepts and relationships, thereby enabling knowledge sharing. So we kind of go from these very philosophical concepts to a really practical application, right? So part of the challenges in biology has always been, and in science in general, has always been to come to a common language. And this is one of the reasons why the taxonomic naming scale was so important. Once we come to a common understanding, a common vocabulary, a controlled and consistent vocabulary, it's so much easier to share information. And this is one of the places where ontology has really shined. So what does that mean? Well, from a practical view, ontology is the representation of something we know about. So ontologies, they consist of representations of things that are detectable or directly observable. And often they are also going to tell us the relationships between those things. So if we saw a wheel, we would say, a wheel or a tire is a part of a car. That is an ontology. That is a relationship between those two things. I defined what something is, a tire, and I defined a car, and I defined their relationship. So what ontologies do, right, is they divide up human knowledge into a clean set of categories. The example here is you could try to figure out where in Netflix you might want to try to find a suspenseful black comedy, right? So I just used 3 different ways of defining what this thing is that I'm looking for, right? How do we take all of our human knowledge and put those into clean categories that we might all easily understand what a suspenseful black comedy is, but being able to find that in a sea of other entertainment is really challenging. So, another question you could ask is this, are movies part of art or entertainment, right? Some people would imply that those are two possibly not necessarily uncomplementary things, but not the same. If we think about just sort of what's in a name, right? Like even when we find a way in a common vocabulary for describing things, sometimes those definitions change dramatically depending upon context. So, what is a cell? Well, a cell could be this. You've got a battery cell. This is an old cell phone, right? A cell. Uh, not even a flip phone. This one. This is a cell, right? A prison cell. We've got a cell of an Excel spreadsheet, a really old Excel spreadsheet. And, of course, this is a cell. We've got, the classic biological context of what a cell is. Um, so it's important that we realize that, humans have done this time and time again, but the same name can often be used to describe different concepts. So, if I'm looking at this set of reactions, of biological processes, and different components that are involved, what would I actually call this, right? So there's a lot of things that I could call this. This process by which I'm making glucose, right? I could call that glucose synthesis. I could call it glucose biosynthesis. I could call it glucose formation. I could call it glucose anabolism. I could call it gluconeogenesis. All of these refer to the process of making glucose from a simpler component, and they've all been used in the scientific literature, right? And so how do we know that we're talking about the same thing? How do we know that we're referring to the same pieces? As we talk about things going on in different species, the more we can identify what is the same and what is different, it's going to really help us understand the evolution and complexity of life. So one thing to hone in on too is that while the same name can be used to describe different concepts, a concept can be used to describe different names. Seems kind of like I'm talking in circles here, but I hope that you can see where I'm getting at. So when we have this case where the same name can provide different concepts, the same concept can be described by different names. Concept can be described using different names, right? What we end up is that comparisons get to be really difficult, right? Especially across species and especially across databases where they all might function under different rules and standards. And those aren't due to sort of necessarily like smart choices that scientists have made over time. They're heavily influenced by the history of those databases and the history of the people who developed them and worked on them. So, specifically though, we're talking about genome annotation. And when we talk about genome annotation, one of the big things that we're going to talk about is the gene ontology. So a part of the solution is the gene ontology. It's a part of the solution. It's not the only piece of the solution. What is the gene ontology? Well, it's an annotation system. So it's a way of establishing the information and relationships that are associated with things that we find in the genome. Provides a controlled vocabulary that we can apply across all organisms. And it's used to describe, typically it's used to describe gene products, proteins and RNA in any organism. So how does the gene ontology, or what we often refer to as the GO, work? Here we want to focus on what's the information that we actually might want to capture about a gene product. Typically, there might be three types of information that we're going to store in the gene ontology. What does a gene product do? Why does it perform these activities? And where does it act? And so if we think about that, what happens is we have some sort of gene product. We think about what does it do? This is defined as molecular function. We talk about what process it is involved in. This is the biological process that it's contributing to. And where does it act? We refer to this as cellular components, where in the cell we would find this gene product, right? And these are the three main ontologies, the three main branches of the GO or gene ontology. So as we think about the structure of the gene ontology, we have molecular function. As I mentioned before, this is the elemental activity or task that an entity is undertaking. We also have biological processes. These are the biological goals or objectives of a genetic product or gene product. So some examples here include things like purine metabolism and mitosis, whereas molecular functions would be like carbohydrate bonding or ATPase activity. And then finally, we have cellular components, and this is the location or complex in which we would find this gene product. And so this is pretty straightforward, just a where kind of piece of information. So it's in the nucleus or on a telomere or part of the DNA polymerase II holoenzyme, these kinds of things. So, if we think about a gene product, say we had a gene product, which was a hammer, we could think about what its function was and what its process was. And so we could say, a hammer, part of it, one of its functions is to drive nail into wood, and the process that would be involved is carpentry. We could say you could also use it to drive a stake into soil, right? And then it would be involved in a different process, not necessarily carpentry, but in this case, gardening. We could also use a hammer to smash a roach, right? And then that would be a different process. That would be pest control. And we could also use it as a juggling object, right? And that would be entertainment. So you can see how just one entity can be defined in many different ways, in different functions and different processes. We could think about the cellular components where a genome product acts. It's pretty straightforward. For instance, we could say, is it on the inner membrane? Is it on the outer membrane? Is it in the crystalline matrix? Or is it just throughout the mitochondria? If we think about the mitochondrial membrane, this is what the Gene Ontology looks for it. We can see all the different things that we could associate with the mitochondrial membrane. So the mitochondrial membrane is associated with an organelle membrane. It's also part of the mitochondria. It is a membrane in general. We find it in the cell. Right? These are all locations associated with the mitochondrial membrane. This is also a nice demonstration, and we'll talk about this a little bit more, about how the gene ontology is actually a hierarchical structure. So we go from sort of the most general components up here at the top, most general descriptions, down to the most specific. Biological processes, we talked about this before, but gluconeogenesis, or however glucose biosynthesis, this whole process, we can see it forms a sort of a network of things, of components that are contributing to this one biological process. There's more than one thing, right? We might look at something like gluconeogenesis, and again, gluconeogenesis is the specific term down here, but we can see that there's a variety of different biological process pathways that could be involved in it. We could be looking at alcohol metabolism. We could be looking at carbohydrate metabolism. We could be looking at the generation of precursors for metabolites or energies, right? We could be thinking about these from carbohydrate biosynthesis or carboxylic acid metabolism, right? So there are different ways that we can track all the way down to this very specific biological process of gluconeogenesis. Molecular functions, these refer to a single activity. So this is not a network of things anymore. This is a single activity, reaction, or activity, not a gene product. So a gene product could actually have multiple molecular functions, right? A lot of gene products are don't have just one specific function. But it is a set of a bunch of different functions that actually will make up a biological process. So one gene product could have multiple functions, and then you could have multiple gene products with using their various functions to actually make up a biological process. So if we think about our previous presentation of the gluconeogenesis pathway, right? Each one of these little spots in this pathway all refer to a different molecular function, right? A different enzyme, which is creating an intermediate step on our way from going from phosphoenolpyruvate to glucose 6-phosphate, and ultimately to glucose. So another example of this from a biological perspective is we think about fermentation. We have sort of all of our molecular functions identified here in the fermentation pipeline, where we might be moving from propionate all the way down to we would start with pyruvate, and then we could move either to lactate or propionate, or we could start with pyruvate and move down to alcohol production, right, and producing ethanol through fermentation. We have our biological processes that these are involved with. So while it's the same starting point in pyruvate, and we have these different molecular functions, they lead us to different biological processes. So here we have the lactic acid, propionic acid fermentation we see in sort of like the creation of dairy products and sauerkraut. And over here we've got alcoholic fermentation that helps make our beer and our wine and things like that. And finally, we have cellular components over here on this side, right? We've got the fact that this process over here is dominated by reactions that occur in bacterium, where over here it's mostly yeasts which are driving this process. So the actual process that we're looking at are happening in different places in these cells as well. If we actually look at what an entry in the geo looks like, it usually has a term associated with it, some sort of ID, a definition, some information on when it was, where that reference came from, and then often some comments associated with it. Ontologies typically will include a vocabulary of terms or names of concepts, definitions, and then logical relationships to each other. Right, so this is a structure of how any ontology works. So if we think of an example of how the gene ontology works, right, we could have these types. We could have chromosomes, nuclear chromosomes, nucleuses, and organelles. And each one of them is actually going to have a relationship to these other things. So for instance, a nuclear chromosome, right, is a type of chromosome, right, but it's also we could find it's a type of chromosome. We can also find it in the nucleus, and the nucleus is an organelle. So notice that there's this structure and this connection. This is another graph, right? We've talked a lot about graphs in this class. And what we can see is that ontologies are typically represented by these graphs, and the nodes of this graph are going to be the terms of the ontology, whereas the edges are the relationships between the concepts. Okay, so the nodes are the terms, the edges are the relationships. There are basically these parent-child relationships throughout the gene ontology. A child is a subset or instance of these parental elements. And so we can see this, for example, for chromosome, right? There's chromosome is the general term, the parent term, and the child terms are cytoplasmic chromosome, mitochondrial chromosome, nuclear chromosome, plasmid chromosome, right? All subsets of chromosomes. Also, gene ontologies generally are presented, and you've seen a couple examples of this already, but they're presented as a hierarchical direct acyclic graph, or a DAG. So acyclical, they only sort of flow in one direction, if we want to think of them that way. Terms can have more than one parent, so it's not like you can only have one parent. You can have multiple parents, and 0, 1, or more children. So those are the options for what this direct acyclic graph looks like. And all the terms are basically linked by one of two relationships. It's either an is-a or a part of. So in this case, if we went back to the original case of the nuclear chromosome, we could see, the path from a child term to its top-level parent. We can assign, in this case, all these red arrows, right? These are is-as, so a nuclear chromosome is a chromosome, a nucleus is a organelle, right? But where it's orange, we say a nuclear chromosome is a part of the nucleus. One thing that must be true when we apply this direct acyclic graph in our gene ontology is it must adhere to a true path rule. And so this states that the path from a child term all the way up to its top level parent must always be true. So that's one of the rules of an ontology. So these relationships that we're looking at must always be true in all cases. And if they're not, then we have to redefine the ontology. And so this is just another example of this direct acyclic graph, right? So we could think about this, tissue regeneration. We could say is a or is a part, right? Is a part of wound healing, right? We have organogenesis is a part of organ development. All of these different components have the types of relationships. So every time we see a black to the parent term, so we could say, tissue generation is regeneration, right? Tissue regeneration is growth, right? appendage morphogenesis is morphogenesis, right? These are all the ways that we can define these different relationships. So a couple no-go areas, parts that are not in the GO. The GO covers normal functions and processes, right? So you're not going to find anything in the Gene Ontology related to pathological processes and experimental conditions. This information will be stored in a different database in a different place. You're also not going to find evolutionary relationships here. There is no implied evolutionary relationship anywhere in the GO. It's independent of it. The system works independent of an evolutionary context. There are no specific gene products. We're not concerned with, a specific protein or any of these things, right? We're not interested in like metabolites and things of that sort. We're thinking of biological processes, molecular functions, and cellular content and cellular components. And this is not a system of nomenclature, right? So it's not like a traditional system. This is an ontology. So the relationships among these things are what's really important. So some additional points. Any gene product, again, can have several functions, cellular locations, and be involved in many processes. So don't think it's only going to be one. Annotations of gene products to one ontology is independent from its annotation to other ontologies. So they're not necessarily related to each other, annotations. Annotations are only terms reflecting the normal activity or location of an entity, right? So they don't look at these exceptional cases. And there are lots of usages of unknown geoterms, so things where we actually don't know what they do. But that is actually a way of naming something, right? So when we call something unknown, it's different than if we say that it's unannotated. So unknown is used by a curator to determine that there is no existing literature to support an annotation, but it does mean that a curator has actually looked at it, versus unannotated, where there has been no annotation at all. So no one's ever looked at it before. And so, like I said, if we think about the Gene Ontology, it kind of breaks up into these large different groups. And how do we place things into different groups? Well, a lot of times we're going to have to use various types of evidence. And one of the amazing things about this is really, there's a lot of curation, a manual curation that goes into this process, right? This is not an automated process per se. This is something where scientists actually go in and spend time correcting the roles and relationships associated with these entities. So there's a bunch of different ways that we would establish what an annotation is. There are various experimental evidence codes that are associated with them. And, anytime we talk about evidence, they might have different weights. Like some things you might think is having better evidence for genome annotation. Other things you might think of or not as good evidence. And so I'm going to go through a couple of these quickly just to show you, and you can see like the level of confidence you might get out of different types of evidence, right? So if we think about some of these experimental evidence categories, like the inferred direct assay, right? This is a direct assay of function, process, or component, right? So this is, cell fractionation, enzyme assay, in vitro reconstitution, like immunofluorescence. These are like, I see this in this place doing this thing, right? That's pretty good evidence. These experimental codes, you're going to have a lot of direct evidence in these cases, right? I'm going to infer function from a mutant, from an RNA interference experiment or a knockout or something like that, right? And this is, again, a very strong direct experimental result. I could also look at genetic interactions, right? Like how much two genes might actually interact with each other. I could show that they're physically proximate to each other and involved with each other through co-purification or protein binding or co-immunoprecipitation. I could show that they're expressed, right? That the location or timing of gene expression is associated with a specific function or activity. I could also look and see whether or not they are really similar, right? So now I'm starting to get into things that are moving away from the experimental results to more computational results. So here I can look at these sequences. Like I say, well, is this sequence the same as this other sequence? Are the domains the same? Are their structures the same? Maybe they're doing the same function. I can look at reviewed computational analyses. So these are non-sequence-based computational analyses. Again, just things where we're looking at maybe how similar to genomes are in terms of their predicted interactions and things of that sort. Text mining, which isn't based on any sort of functional knowledge. I could infer things by genomic context, like where in the chromosome these things might be located, or if they're associated with specific expressed groups like operons and bacteria. And I can start looking start moving away from some of this computational information, even start getting into stuff that's at the edge of either computational or sort of reported by specific authors. So in this case, am I looking at electronic annotations? Something where I have a blast hit or an interpro to go map, right? I could look at the things that authors write in their papers. That's also information. Now, I might not have as much confidence in this information, but this also might be a good source for finding things, especially for older things that may not be maybe the best place where I find just somebody who's done an experiment on it but hasn't just reports it in an author statement. There's non-traceable author statements, right? So statements in a paper that cannot be traced to another publication. So an author might say something but actually doesn't have a good resource for it, but says they've seen it. And then there's just cases too where like someone might just say something and there's no biological data available to support it. So often this is something where like a curator has looked into something but hasn't found anything. There are things also where the curator might look into something and they don't find evidence, but they think that they can infer something. So, a transcription factor we would expect to find, say, being in a nucleus. So a lot of times when you actually might want to think about when you find things in the gene ontology, like how much faith do you have that they're actually correct? Is the ontology good? Is it accurate? And so you want to ask yourself, well, where did this evidence come from? What is the experiment that shows that this thing does what it does? And there is this whole sort of decision tree, right, for letting you know like what goes in what category and how it's used and which ones are. There are systems in place too for ranking these different evidence codes for which ones people think are the most reliable. A good tool for annotating things in general, I think, is a tool called OmicsBox. It's one of the ones I like. There's a bunch of them out there for using the Gene Ontology. David is another really popular one. But I like OmicsBox. And used to be known as Blast2Go, so Blast2Go, right? And what I like about it is like it takes all these different annotation tools and it brings them all together in one place. So Gene Ontology, KEGG maps, Interpro, enzyme codes, right? And basically it typically works by you take something unknown, you blast it, from the blast result, you figure out if any of these results map to GO resources, and if they do, you run them through sort of a filtering process to see whether or not they're a good enough match and the evidence is strong enough to say that the annotation would actually be that you could associate an annotation with this entity. So it's a pretty straightforward process. It takes about 3 steps. You blast, you map, and you annotate. And the application of this annotation rule, again, shows sort of like if we're going to call something one thing, we look at the highest hit that we find, the best result we get from our blast results, and we look and say, how similar are these top results to each other? Are they identifying the same gene ontology categories? And if so, we can use that to identify whether or not we would support that ontology being associated with this DNA or protein sequence. And also we're going to look at each of these things that it hits, and we're going to figure out how good the evidence is. And from that, we'll be able to generate this annotation score. So that's generally how the ontology works. Now let's think about how we actually identify things by sequence similarity. Again, we're basing this off of homology. We've talked a lot about BLAST, so I'm not going to go into BLAST because you guys basically understand how BLAST works, right? But I am going to talk about some other examples. So one of the classic ways that we might identify, and another classic way we might identify something based upon functional annotation is using a sequence-based motif. And so in this case, what we're really interested in doing is trying to find known domains within a query sequence using a sequence domain or motif search, where we try to find something where we know that this sequence or domain or motif is associated with some known function. The principal tool we often use in this case is based off of a hidden Markov model approach, where we're trying to identify pattern. And so in these cases, we're not necessarily trying to match a specific sequence and do a sequence alignment. We're looking positionally and trying to see whether or not the progression along a sequence matches a pattern that we would associate with another known sequence domain or function. PFAM, or the protein family database, is one of the main, another one of the databases you could use for this. InterProScan, which I mentioned before, is another one. And this is used for identifying protein domains and families by finding specific domain architectures in a sequence. InterProScan does a similar thing, identifying protein families, domains, and functional sites. And within InterPro, there's a whole bunch of different tools: ProSite, Panther, Superfamily, all used to identify where these domains are. And most of them using a hidden Markov model approach to do this kind of identification. So just to remind you, right, what are Markov models and hidden Markov models? Right, when we talk about Markov models, we'll use an example of weather prediction here to drive home the point. But say you have 3 types of weather: sunny, rainy, and foggy, right? And you want to guess what the weather is going to be like tomorrow based on a history of observations of the weather, right? So you might say, well, I want to know what the weather is going to be tomorrow based upon given what the weather has been in the past, right? So I'm going to estimate some kind of probability of what the weather is going to be like tomorrow based upon what the weather's been in the past. So here you could say, okay, well, what's the probability that, tomorrow the weather is going to be rainy based upon the fact that today it was foggy, yesterday it was sunny, and the day before that it was sunny. And if we had a case where we were even just looking, n is equal to 5, where we're looking 4 days back in the past and trying to predict what the day what tomorrow was going to be, we'd have about 243 possible past histories that we'd have to look at. And that's a lot, right? That's a lot of different paths to explore. And one of the nice things we can do is we can take a simplifying assumption called the Markovian assumption, right? And this is basically saying, like, I don't need to think about the complete history here. I just need to look at today. So if I look at today, it doesn't really matter if the previous 2 days were sunny. I need to know that today was foggy. That's going to be as good a predictor of what tomorrow is going to be as anything else. So the Markov model, right, is going to take that approach. So it's going to say, okay, I'm going to look at, if I'm going to figure out the probability of what something is going to be, what the weather is going to be today, I don't need to think about all these other sorts of things. I just need to look at one previous time step, okay? So we could have some probability. So based upon today's weather, like, we could figure out, well, what's the probability of tomorrow's weather? We could use data to describe this, right? So if it's sunny, what's the probability that tomorrow's going to be sunny? If it's rainy today, what's the probability tomorrow that's going to be sunny? If it's foggy today, what's the probability that tomorrow's going to be foggy, right? And once we have this information, we can plug it into one of these one of these graphs that we talked about and showed earlier, right? And we can see that we have probabilities of staying within the specific groups and probabilities of moving between specific groups. So if I answer the question, given that today is sunny, what's the probability that tomorrow is sunny and the day after is rainy, right? This is saying, okay, well, what's the probability that I'm going to be able to predict the next 2 days? So today's sunny. What's the probability that tomorrow's sunny and the day after is rainy, right? And Well, what do I need to know in order to make this prediction? Right? Well, I can take this prediction and say, okay, well, I can break it up to what's the probability that it's going to be rainy in 2 days, given that it's sunny tomorrow and sunny today, times the probability that tomorrow is sunny, given that today is sunny, right? And that's what this probability will come to be equal to. And what we can see here is that we can then simplify this with a Markovian simplification assumption, right? And just say what's the probability that it's going to be rainy tomorrow or on the third day, that based upon the fact that tomorrow is going to be sunny, times the probability that tomorrow is sunny given that today is sunny. And we can calculate those two, right? Probability that it's going to be rainy versus based upon the previous day is sunny, and the probability that it's going to be sunny based upon the previous day being sunny. So it ends up being 0.05 times 0.8. We can then start to look at a bunch of different options, right? So maybe, what's the probability that today is, what's the probability that today, given that today is foggy, what's the probability that it's going to be rainy in 2 days, right? And here we could say, yeah, so it's foggy today, and in 2 days it's going to be rainy. There's kind of 3 different ways it could get to that point, right? Like it could stay foggy, then go rainy. It could go foggy to rainy to rainy. It could go foggy to sunny to rainy, right? So in actuality, what I know is what's the probability that it's going to be rainy. I'm going to ignore this middle day, given that today is foggy, right? So it's probably going to rain in 2 days, given that today is foggy. And so if I actually want to calculate that, right, I start having to generate these other probabilities. So I used to have to use my additive principle to say, okay, what's the probability that day 2 is foggy and day 3 is rainy, given that day 1 is foggy, right? And I have to add up this other path and this other path. So see, these are the 3 different paths that I have to add up. And once I create this entity, right, I can start to break them apart, each of these entities, into 2 different probabilities and parse them out. So here I can just say, okay, well, I can figure out what's this probability here. I can break it up into what's the probability that it's going to be rainy in 2 days from now, given that tomorrow is foggy. What's the probability that it's going to be foggy tomorrow, given that today is foggy. I can add them all up. And this is how I can figure out what this probability is. And again, this is just an application of the Markovian model. So I'm just trying to get us to see again. We're talking about pathways through this graph. So hidden Markov models change it a little bit more because now we're not going to be able to directly observe the weather. So why this applies well to sequences is, if you can think about sequences, we will never see the intermediate steps between a lot of the entities that we're concerned about. Whether it's if we want to take the evolutionary example, we're never going to know what the ancestral sequences of these shared co-ancestors were that have the same sequence that are now different species, because that doesn't exist anymore. So how can we say something about data when we can't actually directly observe it? So in this case, still thinking about the weather, suppose you're in a locked room for several days and you were asked about the weather outside. Only piece of evidence you have is whether a person comes into the room carrying your daily meal, who comes in the room carrying your daily meal is carrying an umbrella or not. So in this case, what we know is that if it's sunny outside, the probability that you're going to carry an umbrella is 10%. If it's rainy outside, the probability that someone's going to have an umbrella is 80%. And if it's foggy outside, the probability that someone's going to have an umbrella is 30%, right? And what we can do here is we can now base our probability of the weather, right? We can say, well, now it's not based upon me actually observing the weather of the previous day or anything like that. It's based on me observing whether or not someone's got an umbrella or not. So here we can say, what's the probability of the umbrella given the weather times the probability of the weather divided by the probability of having an umbrella. And as we start to expand this out, what you can understand is if your caretaker has a prior probability on any given day of carrying an umbrella is random, it's 50%. What's the probability that the second day after that is going to be rainy? So here in this case, what we can actually define this as is saying, okay, what's the probability that tomorrow is going to be rainy, given that today is sunny and the caretaker had an umbrella, right? So we have to figure out a whole bunch of different probabilities in this case. So we can define that probability then as what's the probability that 2 days from now is going to be rainy, given that given that today is sunny. What is the intersection between the probability that tomorrow is going to be, that the second day is going to be rainy, given that tomorrow is sunny, and given that the caretaker is carrying an umbrella, divided by the probability that today is sunny, given that the caretaker is carrying an umbrella. In this case, it's important to understand that whether or not the caretaker carries an umbrella and tomorrow being sunny is two completely independent things, right? So we can remove the fact that, whether or not tomorrow is sunny, we can remove the fact of there being a requirement of the caretaker carrying an umbrella from the denominator of this equation. We can then use Bayes' rule, right, to redefine some of these probabilities. So we can redefine the probability that the second day is going to be rainy and that tomorrow is going to be sunny, given that the probability of carrying an umbrella is, given that the caretaker was carrying an umbrella, to the probability that the caretaker is carrying an umbrella, given that tomorrow is going to be sunny and the second day is going to be rainy, times the probability that the second day is rainy and tomorrow is going to be sunny, divided by the probability that tomorrow is going to be sunny and the caretaker is carrying an umbrella. So we can then apply that's our Bayesian rule, right? We can convert this using Bayes' rule. We can then take our Markovian assumption and say, well, it doesn't really matter what's happening tomorrow when I care about what happens in 2 days, right? So I can cut out this bit of information there. That's not really that important. And then I can also take this assumption here. So anytime, right, I have the intersection of A and B, I can redefine that as the probability of A given B times the probability of B, right? So I can take this right here, which is the intersection between these two conditions, and say, well, now I can define it as the probability that it's going to be rainy, given that in 2 days, given that tomorrow is going to be sunny, times the probability that tomorrow is going to be sunny. Now I can cancel out these two sunnies here and here, right? And I end up with the probability that if the caretaker is carrying an umbrella, given that it's rainy, right here, times the probability that it's going to be rainy in 2 days, given that tomorrow is going to be sunny, divided by the probability that a caretaker carries an umbrella. So in this case, I know that the probability the caretaker carrying an umbrella, if it's rainy, is 0.8, right? So probability of having an umbrella if it's rainy is 0.8. I know that the probability of it going from sunny to rainy, right, is 0.05. And then I know that the probability that the caretaker is going to be carrying an umbrella on any day is 0.5. So I can figure out what the probability is that the second day is going to be rainy, assuming that the caretaker has a probability of 0.5. So that's right. Just think about that. Look at all the information that I leave out. I can leave out and still come up with a probability of what this answer is based upon not even looking at the weather, but just looking at an umbrella. Okay, whether or not the guy's carrying an umbrella. So that's how Markovian models work, right? They're basically giving us a series of probabilities that can tell you how likely a particular sequence is supposed to have descended from an ancestral sequence, and vice versa, without actually looking at that ancestral sequence. Like, we'll never know what that sequence is. It's like a board game with rules and an objective, right? That's how we don't like to think about this Markovian model. It's like a game. We've got rules and an objective. And the objective is take any given sequence and find how likely it is to come from an ancestral sequence. That is, obtain the highest possible score, right? What's the best possible score? Well, ranking up the fewest possible penalties, right? So what's the best match in this case? And alternatively, you can use a single sequence. You can say a single sequence and say, well, how does this one sequence fit our Markovian model? So once we build our model, how well can we now say this sequence fits our model? And what you end up with is a pretty drawn out a game here. So these are all the rules of the game. I'm not going to beleaguer every single one of these rules, but basically, every square gives you a letter. DNA of 4 letters, proteins you have 20 letters, and for each one of these, right, there's some sort of score attached to it, either between 0 and 1, and what you're trying to do is get the best score, right? And the longer you stay on any one square, the better your model is. So each time you move to a new square, there's going to be some kind of penalty because that's going to cause you to lose points, to lose score. And so what happens is you end up with a really simple version of this where we just have 2 kinds of cases. So here we would say, okay, we have square 1 where we could have A's and T's, and square 2 where we have G's and C's, right? What we could do is we could use this and try to figure out at which point, am I going to hop from one square to the next? And you could think about this as being the same case as that graph, right? At what point do I go from sunny to rainy? It's a high probability I want to stay in sunny for a long time, but at some point I may want to go to rainy, transition to the next position. So that's how we end up having this position-specific graph that we're going to try to navigate through. And that's how these hidden Markov models work, right? So it looks like, if we know what the board looks like, but not the path, right? We can figure out what's the best possible score we can get. We can figure out what's the best possible path we can travel, right? What we can also eventually do is we can also build, maybe even build a better board game, a better board to play our specific game on. So a better model in general, right? But because we're able to identify this best score and this best path, right? We can use that to say, okay, I can look at characteristics of proteins and say, hey, does this thing look like other kinds of proteins? Does the best path, match this board? Which board does this path match? Which score does this path is highest for what board? And so I can look and say, hey, do I see domains that are associated with signal transduction? Do I see domains that are associated with acid binding? Do I see domains associated with cytoskeleton or voltage gate? Do I see domains associated with immunoglobulins or calcium binding proteins, right? And by finding these different boards, by finding these different path graphs that are associated with the sequence, that's how I can functionally identify what these things are. So that's a discussion, the end of our discussion on Functional based annotations, I'm not going to go into BLAST. We already know how BLAST works, but going a little detail about how PFAM and the GEO work. So hopefully now we've got some background. When we go into class, we try to apply some of these concepts. You guys will have a good familiarity about how they work and how they function and what you can get out of them.